project discusses the application of RNA Aptamer E07 for chemotherapy delivery in pancreatic cancer cells.
So aptamers are a nucleic acid-based platform for specific molecular targeting in biological systems that represents a very promising alternative to antibodies. And so, while antibodies are currently the preferred platform for this, they have several limitations since they're these large, complicated, and sensitive proteins, so they have stability issues, and they're relatively difficult to produce as well. Aptamers, on the other hand, are these small nucleic acid, and they're a lot more robust and tunable, and they're much more easy to produce because you can generate them through this select protocol. And this select protocol is an evolutionary method that allows for easy generation of aptamers to bind to virtually any target. And once you have an aptamer, you can just amplify it, and then you're good to go.
Pancreatic cancer has an extremely low survival rate, and the best available known chemotherapeutic drug, gemcitabine, has limited efficacy due to its toxic effects on non-cancerous tissues. Most pancreatic cancers overexpress epidermal growth factor receptor, which is a transmembrane receptor tyrosine kinase. Its activation leads to cell proliferation, and therefore it is overexpressed in cancers. Researchers hypothesized that targeted delivery of gemcitabine into EGFR-expressing cells by an EGFR-specific aptamer known as E07 could maximize therapeutic effectiveness while simultaneously minimizing toxic side effects due to reduction of uptake into non-cancerous cells.
This study looks at two variations of pancreatic cancer cells in humans that showed that the aptamer gemcitabine polymer was effective in delivering the drug to the targeted cells expressing EGFR and inhibiting growth, which you can see here highlighted in green. And this growth was inhibited with target cells that can occur with drug concentrations less than half of that used without the aptamer. So this inhibition is seen outlined in red without the aptamer. And this, again, will reduce toxicity for other cells in the body. These two types of cancers also showed a similar response to the drug without the aptamer, suggesting that these findings are due to the aptamer's success, not solely to drug sensitivity of the two variants. From these results, we are able to better understand the mechanism for the EGFR pathway in cancer cells. And as you can see here, the polymer of the of the aptamer gemcitabine binds to the EGFR. It is internalized by the cell, degraded, and then the release of that gemcitabine leads to the cell death.
Okay, so overall, this study highlighted the advantages of using aptamers over antibodies. It showed that targeted aptamer-mediated delivery of chemotherapy drugs to cancer cells is indeed possible, and that it's a promising, less toxic cancer treatment technique. This study also showed that RNA aptamers have the potential to maximize effectiveness while also reducing the toxic side effects of chemotherapy by directly targeting cancer cells. Aptamer-mediated delivery of chemotherapeutic agents is a promising treatment technique, not only for pancreatic cancer, a very difficult cancer to treat, but for a wide array of cancer types and stages.